Thank you, Camila, Ruben, for the invitation. I will use this opportunity to introduce you one of these European projects that just started recently, 2 or 3 months ago. And I think it's a very good opportunity to seek for cooperation between the scientists from this project and the LIA MORFUN scientists. So the short name is Ocean Certain. The long name is Ocean Food Web Patrol Climate Effect, Reducing Targeted Uncertainty with an Interactive Network. Here's the motto of the project: creating more certainty about our ocean during climate change. So this is the major things. I mean, you can imagine that, I mean, this guy trying to solve, to save the world, more or less. And it's a teamwork with different scientists. I will show you later all the different institutions which are inside this program. This talk I will divide in 2 parts, split in one. The first one, I will give you an overview about the project, the Ocean Certain project, the structure, the rationale, the goals, and the scope of the project. And the second part, I will show you some examples from the Chilean Patagonia, part of the work we have been done during the last 15 years, more or less, concerning natural climate productivity variability, possible stressors affecting primary production, chlorophyll, and the biological pump or carbon export, together with the food web in this area. I will show you some examples where we find nice results, or at least this is what we thought. But then when the modelers that have to face, all of these global change issues ask us about different aspects, then we realized that there was too much uncertainty than solid knowledge in some aspect, okay? So some general remarks and also some rationale. Of course, we are interested in microbial part of the food web, microplankton, in particular phytoplankton, because they are critical, they play a pivotal role concerning in the regulation of today's carbon cycle. But there is a lot of gaps. That's the main things. So we know the big pictures, but when we start to do some zoom in some of the processes, then we realize that there is a lot of gaps in our knowledge. Well, general things that marine ecosystem from microorganisms up to fish mitigate the accumulation of CO2 in the atmosphere through the activity. For one side, we can talk about the photosynthesis as a process. For the other side, the export of organic matter in terms of different particles like fecal pellets or some other product. But there is a major uncertainties about the importance of these processes and how they are affected by human activity. That's one of the main points. So we cannot cope with all processes in the oceans, but the main aims of Ocean Certain will be to focus on the relevance of the food webs and the biological pump has a pivotal issue that's impinging on the climate change. The project will examine and compare the situation in different ocean areas of the planet. So the target areas are Patagonia, Mediterranean, and Arctic. So you will see why these areas, because they are highly sensitive to global change, but also because in this area there's a group of scientists with enough infrastructure to conduct mesocosmos experiment that I will show you later. The overarching goals, at least from our group, is to improve improvement of the conceptual models describing the integration between food webs, marine export production, this is the biological pump, and the global climate dynamics. Of course, here there is another, the socio-economical scenarios will be part, will be the duty of other groups from different universities, but also is included the social economic part of this topic. Some targeted uncertainties is feedback from food web. We have seen here in some talks that sometime when you conduct some experiment under similar conditions, the pico-eukaryotic picket before and sometime after the increase of temperature, that happens sometimes. Sometimes the flagellate dominate the system, sometimes the diatoms dominate the system. I will show you some results about that. So we finish with nice results, but then when we repeat and we try to explain why, we get in problem and we are in bad shape. The biological pump is another example, sorry. It's another example. The export production, sometimes, we have been used different models. Many of the models use a primary production as a proxy for predict the vertical flux, the export production, let's say. And with the hypothesis that if you have more primary production, you will have more export production. This is the hypothesis. This is what we expect. But in global model, seems to function relatively well. But when we use it in a local perspective in one area, I have more or less something like here of Concepción, the vertical flux of particles together with primary production, which has been measured by Giovanni and some other scientists, and the relationship is really bad. Nothing came out when tried to compare primary production and export production. So it seems that something functions well at the global scale, but not so well at the local scale, at least in this topic. Then we have socioeconomic interaction with food web and the relationship with the food production, which is also part of this project. This is just another example that tried to make some example, because this is rather recent paper with people who have been doing forefront research, like Ulrich Wessel or Ulrich Sommer, or people who have been in charge of the mesocosmos experiment in different countries. This is examples of Germany. And try to predict what happened with the final, competition when you put some community of phytoplankton. And this is the major outcome from this experiment and this project. I took this part from the abstract. Most of these findings are contrary to results from similar experiment conducted with plankton population sampled in spring. The other was in summer, so it was completely different. Well, this is expected. I mean, if not the same season, probably they'll change, revealing large uncertainties in our knowledge of temperature sensitivity of key processes in marine carbon cycling. So this is the main focus, the main point of the project. We have a lot of uncertainties when we are doing experiment work, and the idea is to try to tune all of these results to discuss how we can improve the system in order to give some answer to the people who are eager about information to feed the global model concerning global change. Also, they say that the initial phytoplankton composition when you start some experiment, when you start with the experiment, has a lot of importance of the final result of the experiment. It's not the same if you started with the few diatoms or a lot of diatoms. If you have dinoflagellate or not, everything will change. So this is the major, I would say, the schematic objective, goals, or aims of the project. At this site, concerning all of the global change, you have a lot of sources of variability, or sources of stress, increased CO2 in the atmosphere, ice melting. Of course, all of these are related with alteration in the currents, and also will probably enhance the stratification of the water column day by day. So every day you will have some impact in stratification, temperature, all of these stressors. Nutrients, also macro but also micronutrients, temperature, acidification, changes in light, the oxygen content or dissolved oxygen in the water, everything can be changed because of this. If everything changes, how we can manage to do some experiment and give some results to the people who have to cope with this. In addition to all of this natural or anthropogenic impact, you have all of this anthropogenic impact as well, pollution, overfishing. All of these aspects will have an impact in food web and biogeochemical cycling. And of course, in turn will be in the biological pump. And all of this will impact the socioeconomic as well, because the aquaculture fisheries are connected also with this site here. So this is a tremendous, you see, amount of work, or somebody can say maybe it's too much for a project. And so try to make simple, and this is exactly the same that before. So we have to look for stressors here: oxygen, temperature, acidification, CO2, affected marine food web, and the biological pumps in a scenario of climate change. This here has to do with social economic part, with also are connected in this project. A little bit more about the biological pump. This process is well known. So all our biological oceanography courses, we started to explain the biological pump. But remain great uncertainty. This is a key word here about how this process will be affected by changing climate, social, and environmental conditions. This uncertainty has been problematic in predicting future climate change, and experts and politicians are eager to know a little bit more about that. So we know because of the global change, probably the precipitation regime will change, the freshwater input into the different system, like a Patagonian system, will change. So the question is, how will affect the trophic food web, these stressors, and in turn, how will affect the biological pump? So what we want is to strengthening the pump. You know, we know that the carbon dioxide emission has been increased during the last years. So we would like to know more about how to counteract this climate change, how to improve future climate-related prediction. Can we manage the ocean in a way that strengthens than weakens the biological pump? It's because if we increase the stratification, which seems to be the case, because if we increase temperature, we will heat the surface of the ocean. we will melt the glaciers, and will be more fresh water enter the system. If we strengthen the stratification, probably we will weaken the biological pump. This is a logic, sequence. So how we can revert this? And there is people thinking that geoengineering could be, some years ago, this was an alternative. If we pour a lot of iron in the high nutrient low chlorophyll areas, probably we will stimulate the vertical flux, because also we will stimulate the primary production. This was charted after the Lojafex expedition in 2009, where even though we just put 20,000 of iron sulfate in the water in one eddy, the export was almost nothing, zero. It was completely different, because in this example, it was different to the rest. Even the natural fertilization or the artificial fertilization in previous study ended with a massive sedimentation, but in this case was not. So, was completely discharged as a possibility to manipulate the oceans to strengthen the biological pump artificially. So forget it about that. So the Ocean Certain project has 11 different partners. 8 of the partners belong from the Europe, plus Chile and Australia. And it's a project, will run for 4 years, with an overall budget of 9 million euros for equivalent, which is 72 million Norwegian krone. And these are the partners. You see it from different countries. The only country we have 2 partners is Norway and Chile. And these are. You see, it looks that the different group from different institutions will take care of different aspects also of this project. This is the organization in different working package. Working package 1 is a knowledge generation. Well, in fact, it's data mining and knowledge discovery. This group, first we have to learn the lessons from the past. So the first step is to collect what is the knowledge which has already done, and to learn from this. So this is data mining. Then there is quantified effect and feedback, socioeconomic dynamic, consilience, and integration. All of this group has to provide with information to this, and so on. How to proceed with laboratory and field study to cope with this challenge? We have to see if we want to look into the food web and biological pump, how to proceed in terms of approaches, how would be our stressors that we choose, because they are the most important one. So I put here the intuitive and also the most common strategy for building microbial food web models is that of linking preconceived plankton functional type, PFT, via trophic interaction. That's the box model. Each plankton functional type is represented by 1 or more boxes, state variables, and represent a large set of organisms sharing some property believed by the modeler to be essential. So we can choose or decide that size is a good, property to be studied, which make a lot of sense to me. The size matter a lot in this aspect. Trophic model, substrate utilization, common predator, essential rotting biochemical transformation could be also chosen as a good property to be studied. So let's some alternative. Plankton functional group, if we have different species that seems to have the same niche in the water column, then we have to explain why. The possibility is that they coexist, is the substrate specialization. See, they're two different because they use different things. Very easy. So they do not compete with the same substrate. But the other alternative, killing the winner, could be also possible. In this case, two different species use the same resources. This is more defense specialist, and this is competition specialist. In terms of competition, this will win, always. This is more better competitor than this one. But here, if there is some predator or parasite that are making, predation or prevent if this predator or parasite reduce the competition, because remove this specialist and release the competition with the defense specialist, and both can coexist. So this is inspired in things that he is part of the Ocean Sorting Project, Frede Singstad from the University of Bergen, and he will be in charge of the Mesocosmo experiment. And so we are in the phase of, we have discussing how would be the best strategy to conduct the experiment to give some answer to the people who want to know more about the global change, how will be affected. This is the evolutionary arm race perspective of the pelagic food web, different group. They all of them use inorganic nutrient, but then they specialize. They are more specialists in the collecting the nutrients or the resources from the water column, because they are very small. Maybe they have a large volume to surface area, or some of them try to use the prey try to become more strong with armor, for example. This is quite common, armor. Even the silicate frustules or calcium carbonate plate, everything can help to prevent grazing. But as soon as the prey starts to cover his body with some armor, the predator looks the way to penetrate this armor. Then some, like Diatom, maybe they can use some chemical weapons, like the aldehyde type compound, to reduce the predation or whatever. So you have this is the evolutionary arm race perspective, but this here you can see that there are small, intermediate, and large organisms. So one alternative is to use the control by same size selective predator. It's heterotrophic flagellate has a very strong grazing pressure of this group, ciliate on this group, and mesozooplankton on this group. This, of course, is in an oversimplification of the system, but we try to keep simple when somebody has to do a model. Try to keep simple as soon as possible. But another way to see this is controlled by substrate specialization and requirement for community type plankton functional types. So this is the approaches of top-down or bottom-up, whatever you want. It's possible to do, but we have to discuss what is better to answer the question in the framework of the project. Finally, we go with this. This is conceptualization of the microbial food web. that will form the basis of this project. And shows from one side what we call it probably the microbial food web, and from this side the classical or traditional food web. These all processes are more slow. Here the processes are more fast. And this line would finish in a more strong export of different particles, organic matter, whatever, from this side. I mean, mesoplankton and diatoms is one of the questions that I have. Do we need diatoms and large zooplankton or tunicate to have a significant export? If the answer is yes, then we have to go to a specific type of environment where diatoms and large zooplankton coexist to have a significant biological pump in the system, whatever. But this is more or less the conceptual framework that we have in mind when we will conduct the in the mesocosmos we have to choose which will be the stressors. Until now we have we thought that for one side nutrients, macronutrients and micronutrients like iron could be one of the candidates, and for the other side the top-down control, increasing inside the mesocosmos the numbers of copepods or whatever, how the zooplankton would affect our community inside the mesocosmos. So, but it's an open question. We have to continue discussing these things in the future. These are different possibilities to use mesocosmos in Norway, in the Arctic, and also in the Patagonia. We have used this 1 cubic meter cubitainers. So has been results relatively well, and we can manage to conduct all experiments using this type. This is 1 cubic meter volume. And so we probably will use this during our mesocosmos experiment. Here will be in the Comau fjord, using the facilities of the Winai Marine Research Laboratories in this place. But this will be. Fact, an ecosystem complex relationship between biodiversity and ecosystem functioning. In situ, our systems are extremely complex because it's a mixture, it's an overlap condition between terrestrial and marine systems. Anthropogenic impact also, aquaculture has been increased during the last year in the Patagonia, and the hydropower industry can increase in the next year as well. For the moment, part of that they are just in a steady state, but I think they will be again reactivated all of these dams and different rivers in the Patagonian system as well. These are facts. And global change, long-term changes in hydrological variables, precipitation, stream flow, glacial retreats, everything has been reported during the last years in this area. So what are the impacts on primary production or microplankton composition? This is some of the questions that has to be tackled or during the ocean certain, of course. Patagonia, as I said before, despite a huge amount of new information obtained during the last 2 decades, our prediction modeling capability to cope with an exacerbated global change scenario are extremely limited. So we are in bad shape for answer many of the questions that has been posed by the modelers at the moment. So concerning different issues in front of an uncertain ocean, global change. How would this parameter would react? These are the core, the center of the project. So now the second part, I have some time? Because now I will show you some examples of information we have obtained in Patagonia, and why I will try to focus and say you why we are worried, because I'm not happy with the next with our challenge for the future. I mean, we know that we have a clear seasonal picture of the variability. This is primary production. You see during spring and summer, the productivity of the system increase. No problem. And during autumn and winter, the productivity of the system decrease. So this is really clear seasonal pattern in productivity. Also, this is in time. Now in space, the flux of fresh water that entered the system is also more or less, we can say, there is some areas like within 45 and 50 degrees south that the huge amounts of fresh water enter the system, and with of course with the seasonal variability as well. But this is the area where we have more fresh water enter the system. In the northern part there is some also, but in the southern part close to Magallanes, not too much fresh water in the system, because it's more related with the glacier melting here. But here's the large rivers. All of these rivers like Baker, Pascua, and they have more than 1,000 cubic meters per second, so it's large rivers. But we have some general trends, even though that there is a lot of variability, we can distinguish some general trends from the fjords up to the open or the coastal area, oceanic area. This is the area from Reloncaví. Here is the fjord of Comau, and this is the inner sea of Chiloé. So there is a lot of silicic acid close to the fjord because the silicic acid entered the system in fresh water. So the fresh water is loaded with silicic acid. But most of the rivers are oligotrophic rivers. So the characteristic is oligotrophic, but with a lot of silicic acid. And we will see as well that with a lot of dissolved organic nitrogen. So this is another important part. We have seen the phosphate and nitrate penetrate from the oceanic side from below, because they penetrate together with the subantarctic water. Subantarctic water from below, the fresh water which is less dense. So mix inside. This is a typical characteristic of our rivers, fjords and channels. And bacteria seems to be more or less the same. I don't know why. I mean, if somebody can tell me why the bacteria are so flat sometimes, or they go from 10 up to 4 to 10 up to 6, from the surface to 1,000 meter depth, from the coastal area to the center of the ocean. Well, but this is the real. We have found the bacteria don't too much variability in numbers of bacteria. When I say bacteria, include both bacteria plus archaea, because we cannot distinguish both group. Krill increasing to the open ocean as well. This is lithogenic organic matter, which are very abundant and penetrate with the fresh water into the system. So this is more or less the general pictures from the fjord to the open ocean in our fjords. Here, this was a paper in 2004 from Parakis and Hedin. They collected samples from different rivers in the Patagonia, and they found that if they increase precipitation, increase the dissolved organic nitrogen as well. What's the impact of the dissolved organic nitrogen into the system? How affect the community? If the stream flow decrease or increase with the global change, how this nitrogen will affect the composition of the phytoplankton, for example, or bacteria, whatever. I don't know if somebody knows, please tell me. Because, yeah, this is a general picture made by Toño Cuevas. From all, he took all the information for 5 years of CMAR project or expeditions, in covering from the northern to the southern part of the Patagonia, the whole Patagonia complete. And this is here, this scale shows from the northern to the southern part of the Patagonia and different variables. And there is something that came with really interesting sometimes. You see, when there is a lot of fresh water, because this is salinity, decrease in fresh water here, decrease in fresh water here, coincide with a dominance. This is not absolute number, but this percentage dominance of phytoplasm. So small cell seems to prefer fresh water. I don't know. This is what the information tells us. Except here in the northern part, where this is probably this decrease in salinity is because of the Pueblo River. The Pueblo River is also 1,000 cubic meters per second. It's a huge amount of fresh water. But here the phytoplankton is almost nothing, but there's a lot of microplankton. This was Skeletonema, this was Thalassiosira, this was so what Chetoceros. So this area has something that is not. I mean, here is most of this water maybe come from the glaciers. This is fresh water from river. This water crosses different agriculture areas. So probably the chemical composition, the stoichiometry of the water is completely different with different results. You see, these are the 3 areas which are presently talking about. And this is the information from the Concepción time series station here of Concepción. This is from 2002 to 2007, 5 years of, almost 6 years of information. This is what we collected in our sediment traps, fecal pellets from different groups of the zooplankton and diatoms. But I would try to call your attention to diatoms here. You see, in 2 occasions, the sediment traps was a huge amount of diatoms that were collected in the sediment trap. So it means the flux of diatom to the bottom. And if we try to connect with the water column, at the same time, at the water column, the chlorophyll concentration was relatively high, and this was diatoms. And secondly, if you see the relationship between primary production and chlorophyll was really low. This relationship is a proxy of the physiology of the organisms. So they are in bad shape and they are abundant. 2 conditions necessary to have this mass sedimentation of diatoms. The rest of the year we didn't catch any other of these. So can we predict when will be the sedimentation high? As I told you before, primary production results a very bad proxy. Diatom together with physiological state may be better. well, this is more question than answer in this area. So in the framework of the biological PAM, do functional group and species composition matter? Of course, yes. I mean, in terms of zooplankton, these are fecal pellets from different groups, Euphaucid, appendicularian, and they have very large, compacted fecal pellet. They sink very fast in the water column. They just make a very nice coupling between the surface and the bottom. So it seems to be a good candidate to put it here at the top. Protozoan mini pellet, they are so small, they are remineralized very soon in the water column. They not sink down in the water column. In terms of diatoms, of course, there is also some rank here. At the top is Thalassiocyra, and here is Skeletonema. Skeletonema is usually more abundant in sediment trap, but they are very small. So in terms of carbon, Thalassiocyra are not so abundant, but the carbon content is so high that they win in terms of carbon flux. This is another example of trophic fluxes in the northern Patagonia region. We compare two different areas. So this is the inner sea of Chiloé and the Moraleda channel here. This has a lot of exchange with the oceans. This has a very restricted exchange with the oceans. And as I told you before, in the ocean is the sources of nitrogen and orthophosphate. And what we found here, that even though there is similar primary production, the fluxes of organic matter, the fluxes of energy from the bottom, from the first levels like phytoplankton to the top levels of the food chain is increased. They are very efficient. And here is not efficient. The difference is here is that phytoplankton is composed of large diatoms, and here phytoplankton and nanophytoplankton. Small cells, large cells. So large cells stimulate the flux in the trophic fruit web in these two different areas. So in this case, topography, depth, extension of the Patagonian basin affect the extent of the water exchange between the inner and the outer part of the fjord, determining marine productivity, biomass level, and energy flow through the pelagic food web. This is an impressive paper that, and this is an example of how the constriction sealed, because this was the area with restricted flux of energy to the top. Okay. And you see, if we go from this side to in the inner part of the fjord, the salinity decrease, and also there's different sub-basins inside, and so silt seems to be very important. These silts also in the fjord ecology and the fjord, and this is finally we try to put all the information together to see how function the system. This is the, for example, here in the inner sea of Chiloé during winter and during spring. During winter, we had a lot of bacterial production and very low phytoplankton primary production. The other way around is in the spring. So the system seems to respond very clear seasonally, but when we make zoom again to different processes, we get in big troubles and problems. This is the nitrogen and orthophosphate penetrating the system from below in subantarctic water, and the silicic acid in fresh water from the top. I will skip this because we try to do some experiment with siderophores and with the iron with different results. We use it with the diatoms and the dinoflagellate to see if they are ligands, because the ligands, the complexation within organic matter and iron occur in all this area. So if diatoms or dinoflagellate try to collect this iron, who win? Who has the capacity to do it? Well, we tried to do some experiment, and the other was with different nutrients. In some experiment, dominating Echococcus picoeucaryotes. Probably we have also in our your pet key, yeah, is probably here as well. But we have a lot of uncertainty on this one. And for tomorrow discussions, I put here some menu of questions for discussing tomorrow or today or whatever you want. But this is, for me, one of the weaknesses we have if we want to understand a little bit how to function our system in front of the global change. And that's it. Thank you. I will